Transcription, translation, and genetic coding, AQA biology. Key terms. Pre-mRNA contains introns and exons. Proteon is all of the proteins that an organism expresses. tRNA is used to carry amino acids so they are close to one another. Gene, sequence of bases in DNA which codes for a sequence of amino acids in a polypeptide. Stop codon signals the end of a codon sequence. Start codon signaling the start of a codon sequence. Degenerate, a codon which has multiple codes with the same meaning. Overlapping, each base is only part of one triplet. Universal, triplet code for all amino acids are the same in any living organism. Introns, non-coding DNA. Exons, coding DNA. Splicing, cutting out introns from pre-mRNA. Transcription. Transcription is when DNA helicase unwinds the DNA section for protein synthesis. It breaks hydrogen bonds and then RNA polymerase joins complementary RNA nucleotides together to form pre-mRNA. So pre-mRNA contains introns and exons. Introns make up about 98% of the pre-mRNA strand. And mRNA contains exons. The introns are spliced out and the exons are put together. Prokaryotic cells are a single process due to the free floating DNA. And the direction that it's read in is from 5' to 3'. And this is a little diagram for transcription. So you have your DNA strand, which is then broken. The hydrogen bonds are broken by DNA helicase. Then you have RNA polymerase here, which joins the complementary nucleotides together, RNA nucleotides together, and then this goes down to as pre-mRNA. Pre-mRNA is smaller than mRNA. What I forgot to mention was that because pre-mRNA is smaller, it's able to leave the nucleoplasm easier. So translation, this is a tRNA molecule and it has an open end at the top which is able to bind to where the amino acid, so let's say this is the amino acid, it would bind at this area. And then on the opposite end this is the anticodon. So because here it's CG, wait, let me double check what that is actually. It's CGG as is the anticodon. So the codon that would be on the strand would be GCC, which would be the complementary pair. So translation is basically when mRNA attaches to a ribosome. Codons in mRNA attaches to complementary tRNA molecule carrying the specific amino acid that codon codes for. A peptide bond is formed and ribosomes move forward. The tRNA leaves and is replaced by the next. It continues into a stop codon and the chain is released and then folds into the correct shape. Amino acid activation. tRNA picks up specific amino acids. tRNA can't pick up amino acid unless it's specific and activated. Many ribosomes work on the mRNA strand at once to speed up the process of protein synthesis and forms a polysome. And a polysome is when a cluster of ribosomes held together by a strand of mRNA and it translates this. So let me show you a little example. So you'll have your mRNA strand here. So let's say this area is C, G, C. So you have your tRNA molecule with your anticodon will be complementary. You have your amino acid that's joined at the top. And then you have your ribosome, which we're going to have in a blue color. The ribosome is here. So when the tRNA binds this, the protein joins onto the chain. and then the ribosome moves forward. So the next area is red. So when this area is red and the protein is placed here, oh wait, let's make another color for that protein actually, because you'll get confused between that and the ribosome. So let's say the protein that's formed is now a dark blue color. This will now form a peptide bond with the amino acid. This will now form a peptide bond with the amino acid that will be placed down when the tRNA leaves. So the tRNA will leave, deposit in the protein there, amino acid there, sorry. And as the R ribosome moves forward and the next one is coded for, it's linked by a peptide bond. And this process just continues until it stops. Genetic coding. The number of bases for one amino acid is 3. So 4 to the power of 3 results in 64 bases, which will code for the 20 amino acids needed for basic human life. They're made in a triplet code and there's 44 extras like we've already discussed. The location of exons and introns. They're basically on genes. So you have your exons which code and your introns that do not. Your base sequence changes as a consequence, as new sequences may form due to mutations. And sometimes because the code is degenerate, the same substance is made. However, if not, then it will cause the primary structure to change due to new bonds being put in place. So it will make new folds, new 3D structure, and the substrate won't fit. And the features of a triplet code is that it's degenerate, non-overlapping, and universal. So here you can see the splicing which has occurred when the introns have been removed, making a small molecule, which is mRNA instead of pre-mRNA. So I want to go on to the thing again. So let's say you have a mutation. Say it was before this, a straight blue line. However, it's now turned into this instead of that blue straight line. So because of the change, it will fold into a different tertiary or 3D structure, potentially quaternary. And because it's changed into this molecule instead of what it's supposed to be, the substrate, which is this shape, let's say, can't fit into that gap. Therefore, it's now non-functional. And that's all it really is. I just wanted to go over that again because I didn't think I explained it the best. Now, that's all there is for this unit, and the next unit will be about meiosis and genetic diversity. So if you like this video, then please like and subscribe, and I'll see you on the next one.